In the Mexican capital's new international airport, an unexpected number of mammoth bones have been discovered. At least 110 individual mammoths have been unearthed since the somewhat controversial construction of the airport began in 2019. But now scientists have analyzed DNA from some of those fossils and found that they represent a previously unknown type of mammoth that split from other North American mammoths much more recently than expected. Before we get into the genetics of it all, let's talk about the site itself. The airport, known as the Felipe Angeles International Airport, is located northeast of Mexico City near the former Lake Zoltocan during the last ice age. The valley was a rich ecosystem inhabited by a diversity of prehistoric animals, including a large number of mammoths, and now their bones are turning up in the foundation for a new highway and the airport itself. The majority of the mammoth remains come from a single area called the Puerco Run. Between 2016 and 2019, 318 specimens from 75 individuals were excavated from here. A little over half of these remains belong to mammoths. While the site was being excavated, paleontologists assumed that these mammoths would be genetically the same or very similar to the Columbian mammoth, the only species of mammoth native to North and Central America. The other species of mammoth found in North America is the woolly mammoth, which clearly migrated from the Eurasian continent and has been mostly found in Canada and the United States. But the origins of the Columbian mammoth are less clear. Past research suggests that it may have risen from the hybridization between woolly mammoths and another Eurasian species, the steppe mammoth, sometime around 800,000 to 400,000 years ago. It seemed likely that the mammoths found in Mexico would be the same as these, but a new study, published in the journal Science, tells a different story. The authors of the new paper analyzed the mitochondrial DNA of the mammoths found during the construction of the airport, as well as samples from mammoths excavated between 2016 and 2019 at the Toltec site. Mitochondrial DNA is found in the cell's mitochondria, the powerhouse of the cell, and is separate from the main nuclear DNA. Most often, extracting usable DNA for analysis in the tropics is difficult since DNA doesn't typically fare well in warmer climates. This is in contrast with the many woolly and steppe mammoth DNA samples found in the far north. Therefore, most prior Columbian and mammoth DNA came from fossils found in more northerly regions of North America. Using mitochondrial DNA instead of nuclear DNA was a way around this problem, although only the maternal lineage could be studied this way, which may not reflect the nuclear genome diversity. The study revealed a distinct mitochondrial lineage of the Mexican mammoths, referred to as clade 1G, separate from other North American mammoths. There were also further groupings within the Mexican group. The researchers say that the groupings within the Mexican group showed deeper genetic divergence than expected. The divergence within clade 1G was as great as the divergence between other major North American mammoth lineages. The study authors offer two explanations for these results. First, there could have been structure at the mitochondrial level in the ancestral population of M. primigenius before the hybridization event with the M. trugontheri-like Krestovka lineage that gave rise to M. columbi. Second, this pattern could indicate different M. primigenius parental populations mixing at different times, with a similar M. trugontheri-like Krestovka lineage giving rise to divergent M. columbi metagenomes. The study authors note that they lean toward the first explanation being the correct one. The researchers also used radiocarbon dating to determine the ages of the fossils, which suggested that these divergent lineages coexisted during the late Pleistocene, between 40,000 to 12,700 years ago, so the genetic makeup of these mammoths is more complex than researchers expected, and the discovery shows that more work needs to be done to fully understand their history. The authors conclude this study emphasizes the importance of recovering ancient genomic data from wider geographical ranges in order to fully understand the evolutionary trajectory of extinct species and demonstrates the feasibility of recovering DNA from late Pleistocene samples from tropical latitudes.